and water move across large distances in plants. However, in plants, the movement of food and water also takes place across short distances. That is, within a cell or from one cell to another. This movement of food and water across short distances takes place either passively by diffusion or by active transport. In diffusion, the molecules move in a random manner, generally moving from a region of high concentration to lower concentration. Diffusion is a passive process. It requires no expenditure of energy. It is a slow process, independent of a living system, and can take place in all states: liquid, solid, and gaseous. Did you know that diffusion is the only means of gaseous transport in plants? Gases such as oxygen and carbon dioxide enter and exit stomata via diffusion. The rate of diffusion is affected by several factors such as the size of molecules, their solubility in lipids, and the concentration gradient. Of these three factors, the concentration gradient is a must for diffusion to even take place. As for size, the bigger the molecules, the slower the rate of diffusion. And since all membranes are made up of lipids, lipid-soluble substances diffuse faster. For this same reason, hydrophilic or water-soluble substances find it difficult to diffuse through membranes. In addition to their existing concentration gradient, diffusion of such hydrophilic substances has to be facilitated by special proteins called transport proteins. This type of diffusion, facilitated by proteins without any energy expenditure, is referred to as facilitated transport. The rate of transport in facilitated diffusion is maximum when all transport proteins are used or saturated. Facilitated diffusion has an advantage in that it allows cells to select substances for diffusion. It is, however, sensitive to inhibitor molecules that react with side chains of the transport proteins. Proteins are responsible for creating channels in the membrane, which allows molecules to pass through them. Some of these channels are large enough to allow a variety of molecules to cross over. Some of these channels are always open, while others are controlled. Some transport proteins, called porins, form pores in the outer membranes of cell organelles, such as plastids or mitochondria, to allow molecules of the size of small proteins to pass through. When a molecule binds itself to a porin, it rotates to release the molecules inside the cell. The water channels are made up of 8 different kinds of aquaporins. Some transport proteins allow diffusion if 2 different types of molecules are required to be transported. Based on direction, transport of molecules can be differentiated into 3 types: symport, antiport, and uniport. Both molecules cross the membrane in the same direction in a symport, while they move in opposite directions in an antiport. In uniport, as the name suggests, a single molecule diffuses across a membrane independent of other molecules. Unlike diffusion, active transport requires energy to move molecules against the concentration gradient. It is carried out by membrane proteins that use energy. These proteins, called pumps, can also transport substances from low concentration to high concentration, which is taken to uphill transport. Just like facilitated diffusion, the active transport rate is maximum when all protein transporters are being used or saturated. Also, these proteins are specific to the type of substances they carry across. They are sensitive to inhibitors that react with their protein side chains. Of all the means of transport discussed, both diffusion and facilitated diffusion always take place along a concentration gradient and without the expenditure of any energy, whereas only active transport uses energy. Plants need to be watered. However, have you ever wondered how seeds absorb water in the first place? Seeds absorb water by a special type of diffusion called imbibition, where solids absorb water, causing an increase in their volume. Imbibition is a special type of diffusion where, once again, water movement is along a concentration gradient. For any solid to imbibe a liquid, it must have an affinity for the liquid. Also, the water potential gradient between the absorbent, that is the solid, and the liquid imbibed is essential for imbibition. The water potential gradient comprises two main components, solute potential and pressure potential. To understand the concept of water potential, we must take into account that water molecules possess kinetic energy. They are in rapid and constant motion in both liquid and gaseous form. The greater the concentration of water, the greater its kinetic energy. As a result, it has greater water potential. Water potential is denoted by the Greek symbol psi. It is expressed in pressure units as Pascal or PA. The water potential of pure water at standard temperature and pressure is taken as 0. If a solute is dissolved in pure water, the solution has fewer free water molecules. This decreases its water concentration, resulting in decreased water potential. Therefore, we can easily conclude that pure water has the greatest possible water potential, whereas the water potential of all solutions is less than that of pure water. The magnitude of the lowering of water potential due to the addition of solutes is called solute potential. Solute potential is represented by the letter psi s, and it is always negative. Solute potential becomes increasingly negative with the addition of solutes. When two solutions come into contact, water molecules from the system with higher energy will move to the one with lower energy. That is, movement will take place from the solution with higher water potential to the one with lower water potential. For a solution at atmospheric pressure, its water potential is equal to its solute potential. But if pressure greater than atmospheric pressure is applied to pure water or a solution, it results in an increase in water potential. When water enters a plant cell due to diffusion, it builds up pressure against the cell wall, causing the cell to become turgid. This pressure that is applied by the protoplasm against the cell wall is called pressure potential. Pressure potential, which is denoted by psi p, is usually positive. However, negative pressure potential is critical to water transport up the stem via xylem cells. Hence, it can be stated that the water potential of a cell is a combination of its solute potential and pressure potential. A plant cell is bounded by a cell wall and a cell membrane. It also contains a large central vacuole.